So plan for the day. You might recall there was a survey that I asked you to fill out about 6 weeks ago now on your own awareness of your thinking and the way that you're feeling. And I'd like you to come back to that, and if you're willing, of course, fill it out again. It's 100% anonymous. I'm not collecting your email address in the background or anything like that. Um, and I'm doing this, of course, because I'm trying to find out, at least from the perspective of correlation, not causation, if these check-ins that we're doing are having an impact on on your awareness of what you're thinking and what you're feeling. I get emails, I get chat messages sometimes that say, "You're the only teacher who's doing anything like this." Well, okay, that's fair. Let's find out if it's making a difference. So if you're willing and able, my hope is you can click on that link directly. It'll take you to the survey. And just take a couple moments here at the beginning of class to answer those. I think there's 5 questions. Just let me know if you can't get to it for any reason. Are we ready for the biology? Folks at home, thumbs up. You're good. All right. Thank you if you have submitted your responses to that survey. I appreciate it. Again, I'm just looking to see if there's any sort of impact. Not that I can say it's this that's having the impact, but if there's any sort of correlation between what we're trying to do and your awareness of what you're thinking and what you're feeling. Alrighty. Feels like all 8 of these classes now are in different places. So I wanted to kind of go back a step before we go forward. And you've watched 2 videos now on the structure and function of proteins. So here is a picture of a very simple polypeptide, really the skeleton structure of a polypeptide. What's taking place in this picture? Which answer choice makes the most sense to you right now? Right. Let's see how you're doing. All right. Good. This is indeed a hydrolysis reaction, right? We're watching a very simple polypeptide get split apart into two smaller polypeptides. And this, of course, connects to some of the work we've been doing over the last week or so, right? That we can put molecules together through dehydration synthesis, but we can also take molecules apart through hydrolysis by adding water, by breaking a covalent bond, and splitting the molecule apart into smaller pieces. So, excellent. Well, again, our focus for today is on the structure and function of proteins. And I'm not going to re-lecture on this material. That's ridiculous. You've already watched the videos. But excuse me, a reminder that if we're building a protein, we're building a protein from amino acids. Those are the molecular building blocks. So if we're talking about a fat, the building blocks are fatty acids and glycerol. If we're talking about carbohydrates, the building blocks are monosaccharides like glucose. So while the monomer, the building block, might be different here, the process by which we put those monomers together, it's still going to be dehydration synthesis. So just bring up my pen here for a moment. Notice if we're linking these amino acids together, we've got a carboxyl group at one end. This is why having some knowledge of the functional groups is helpful, but this isn't about memorizing, right? It's about having that table in front of you if you need it. And we've got an amino group for this other amino acid. We're going to link them together with a new bond, a covalent bond. Yes, it has a name. It's a peptide bond. But by linking them together, of course, we're going to produce a molecule of water. So this is really the opposite of what you just saw in the question that you answered. This is not hydrolysis, this is dehydration synthesis. Water is a product, not a reactant. All right, so before we hop into modeling some of these molecules, is there any lingering questions that you have just about the basics that you saw in the videos? Yeah, Ethan.
Could you define a peptide?
Yeah, so a peptide is often a term that's used to describe just a few amino acids that have been put together into a polymer. So when you see polypeptide, it gets a little ambiguous. In general, we mean 3 or more amino acids that have been linked together, but it could hypothetically be thousands of amino acids that have been linked together. In the model that we're going to build here in just a moment, we're going to link 15 amino acids together into a very short polypeptide. Well, what we're going to build here is, in fact, speaking to Ethan's question about what is a peptide, we're going to build a really simple polypeptide. Folks at home, I've got a virtual equivalent for you to explore at your own pace. But suffice it to say that a polypeptide biologically is not the active form of the molecule. A polypeptide really is just a linear chain of amino acids. In order for that molecule to have any functionality, we have to fold it into a unique three-dimensional shape, and that's the protein. The protein has the function. It has the three-dimensional shape. A polypeptide is really just a chain of amino acids that we've put together into a long polymer. We have to fold the molecule in order for it to have function. So that is what we are going to try to model. So folks here in the classroom, I've given you a piece of, we'll do it this way in fact, so everyone can see it, a piece of tomato wire, and then a cup of push pins. So folks at home, this is really what they have in front of them right now. And what I'm going to ask folks here in the classroom to do first is go ahead and insert these push pins into your foam covered wire.
I'm the only one who got it because you like me the best.
Oh, sorry, I'll get you the foam wire in a second. But what you're going to do with these push pins is try to space them out as evenly as you can along the length of this piece of foam covered wire so that what you have looks something like this. Okay. Sorry. I think I like you the best in fact I've given you a somewhat incomplete set of pins. So maybe I'm neutral at best.
I love this thing. I want to buy it. It's so fun.
So it's not going to be perfect. Just as evenly spaced as you can without trying to poke yourself along the way. So folks at home, you can just kind of take a breath here for a moment while they're assembling this. Your setup is not going to be exactly what it needs to be. It's going to be pretty close. Again, to help you do what you need to do. I didn't have enough colored push pins of a particular color to make a complete set, so it's pretty close. Usually you do this with a partner. I know that's a good idea. So this does not need to be perfect. We want to work quickly. We obviously don't have a lot of time together. So if your type A personality is getting in the way, see if you can just take a breath, let that go, get those push pins into the wire as efficiently as you can. I have Band-Aids. So again, what we're trying to assemble here is a polypeptide, a linear chain of amino acids. Just pausing before we move forward. Recall that each amino acid is different from all the other amino acids, and there are 20 in total that can be used to build a protein, by their side chain. Okay, so different side chains have different physical properties, different chemical properties, and that's what makes each amino acid unique. So the push pins in this really simple model are meant to represent different types of amino acids, but really different types of side chains that belong to different amino acids. Okay, so just pausing there before we go forward. Just bring up any questions for anybody at all. So in this model, each push pin has a different chemical property. You can see that in the table on the screen. What you want to do with this polypeptide is model the folding of this polypeptide into a protein, but it's not going to fold in some random stochastic manner. It's going to fold according to a set of really chemical rules. So what are the rules? Well, first, we're making the assumption that our protein is going to be folding, our polypeptide is going to be folding in water, which means all of our hydrophobic non-polar amino acids need to be buried on the inside of our structure. So for you folks, that's going to be the yellow push pins. However you fold your polypeptide, we need the yellow on the inside. Okay. The white push pins are the polar amino acids. Unlike the hydrophobic non-polar amino acids, the hydrophilic polar amino acids need to be on the outside of the protein. Okay. The red and blue push pins, they are negative and positively charged side chains, respectively. They need to be interacting with each other. They're going to form a bond. And then finally, the green push pins are a special type of amino acid called cysteine. They can actually form a covalent bond with each other. They need to be close to one another. Okay. So, folks at home,
The real ones should be next to each other.
here is your protein that I folded for you. Can you drag the dots in a way that you begin to identify the different parts of the protein that have particular chemical properties.
Oh my gosh, I did.
Okay. The only rule, folks, that you have here in the classroom is you can't pull out a push pin and move it someplace else. You've got to fold the polypeptide according to the rules.
But can you go back and do it in student mode so you can see?
Which one do I have? What's the purple one?
Yeah, so your kit isn't perfect. If I remember correctly, the purple is meant to replace one of the yellows because you only have
Oh, so it's just the only inside.
the orange is the same thing. I have used purple and orange to replace two of the missing yellows. Everything else should be good. So these molecules fold into their unique three-dimensional shapes in microseconds. So you should be able to do it on the order of nanoseconds because, you know, you're conscious and intelligent. These molecules are not. He says sarcastic. There is not one correct answer here. You're just trying to practice folding the polypeptide into a three-dimensional structure according to these rules. So for those of you at home, I've got some different questions again on a pre-folded protein that you can answer. So they're on 245, 246, and 247. Trying to make it as equitable as I can. On to the next. If you think you've got something that follows the rules, just turn to a neighbor and show your product to one another. Maybe two neighbors if you're in the middle. Notice what's similar, notice what's different about the models.
That one's just really outside. Yellow on the inside.
You had a chance to
Red ones to go.
show each other what you created? What you folded? What are you noticing? What's similar? What's different? Are all the structures the same?
Okay, so make it similar. There's a yellow right there.
Everybody's protein's the same, right? You've all come up with the answer. We've measured our percent accuracy. We've determined the grade based on the percent accuracy from the normal, from the expected. determined your grade, your status in life, your future relationship status, where you're going to live, how well you're going to live, all that kind of good stuff.
Looks like I'm gonna have a great life.
My hope is that you're noticing that the protein's shapes, the three-dimensional shapes, are different. So what made them different?
The greens are supposed to be together.
Where you put the pins, the sequence of the amino acids with their corresponding side chains, some of which are nonpolar, some of which are polar, some of which are charged.
Sure.
Is this even close?
Yes, it is.
Well, that's just it. There's not one answer.
Yeah, but
Does it fit the rules to the best of your ability?
I'm not sure.
Well,
Yeah,
it
I'm
looks
not
like
sure.
I see the yellow on the inside and the white on the outside. I see the red and the blue pretty close to each other. Maybe the greens are the only ones that aren't. Oh no, I think your finger was just hiding it. I think the greens are pretty close to each other. So that looks like it works. Okay. So again, the goal is to really demonstrate that all the shapes are going to be different because the sequence of amino acids that you inserted, right, that you synthesized, are different. And that's the first major takeaway from this really simple modeling activity. The sequence of amino acids influences the structure of the protein. Okay? All right. So then, my question for everybody, no matter where you are, is this. You folded this protein, whether it was folded for you or you folded it yourself, in an environment that we assume to be aqueous, that we assume to be watery. What would happen if the same polypeptide were to fold in a non-polar environment? Can you take a moment and predict how that polypeptide would have to fold? So again, we made the assumption that when we folded our polypeptide, we folded it in water in an aqueous environment. How do you think that polypeptide would fold if it were to fold in a non-polar environment? Heck, in oil, for example. Oil is a non-polar substance. Or in fat.
What's the difference? What's the difference
Between
between oil and fat?
oil is a type of fat. So there could be solid fat, right? Saturated fat versus unsaturated fat. Okay. Again, can't really move around much in the classroom, but turn to a neighbor and just share your prediction with someone close by. Folks at home, if you'd like, I can put the speaker on mute and you can just share with each other out loud. Take yourself off mute and nobody else will hear you other than the people on Zoom. You are your own breakout room. Whenever you're ready. See what you came up with.
Globule.
Yeah, reversed, right? Reverse would happen. Yeah, the nonpolar need to be exposed to the outside. Outface outwards. Yeah. Outside of the protein. Maybe like a flip-flop. It's a reversal. What was on the inside is now on the outside. What was on the outside is now on the inside. There it is. Inside out. Absolutely. All right. Wonderful. Wonderful. Okay. So you're getting it. That's awesome. Way to go. So I'm going to skip ahead here a little bit. In the second video on protein structure, were you not introduced to Bo the dog? We're told you're going to learn more about Bo when you come to class. So before we go on to Bo, I just again want to just reiterate the importance of the sequence of amino acids, right, along a polypeptide will ultimately determine the structure of the protein that folds from that polypeptide. But it's not the only factor that will influence the shape, the three-dimensional structure of that protein. There's another factor, okay? So to help us see that factor, I found these pictures. I don't know if these dogs actually look like this. But a picture of mommy and daddy, Bo's mommy and daddy, and then his siblings. I don't know if they're male or female, I apologize. But just before we go too much further, what are you noticing about mommy dog and daddy dog compared to Bo and Bo's siblings?
They don't look similar.
Okay. And maybe focus specifically on their fur, their hair. Sorry, right?
Wavy hair.
Dad has wavy hair. Okay. Does mom have wavy hair? No. Okay. How about Bo? Does Bo have hair like dad or hair like mom?
I like both.
Does he?
It's like I feel like he has more fur than mom.
What if I showed you? These are pictures of Bo and his siblings as puppies. These are pictures of Bo and his siblings as young-ish adult dogs. What are you noticing now? Does Bo have fur like mom or dad? Dad? How about his siblings? Like mom? So there's a difference. That's what I'm hoping you're noticing, right? In this very simple example, that Bo's hair is different from his siblings and is more closely related in terms of its, its texture to his father compared to his mother. But we could even argue there's a difference between puppy Bo and older dog Bo.
He glows down.
So when this was observed, this is actually based on real life examples, right? Scientists wanted to know what was responsible for the difference in the hair texture. Hair samples from all of these dogs, mommy, daddy, Bo, and his siblings, were taken, which isn't hard to do, right? Because dogs shed. And the amino acid composition of the hair from all of those dogs was determined, and that information is right here in this table. There's a lot of information here, but if you could just take a moment and scan the table, noticing from left to right, you've got Bo and his parents. And from top to bottom, you've got the different amino acids that make up the protein. It's called keratin, in the hair from all of these dogs. And as you scan across, do you notice any differences? You can actually highlight or circle those differences or put a dot next to those differences as you find them. If you're wondering, ND stands for not determined. It simply means that there was none of that particular amino acid found in the protein, the keratin protein that makes up the hair sample. Let's see what you are noticing. Yeah. Arginine, right? This amino acid arginine, R is the one-letter abbreviation for it. Looks like it's different between puppy bow and adult dog bow. Yeah. Maybe this person thought that that particular amino acid was not found in any of the samples, why they chose to highlight it. Kind of just scan down here. Yeah, same idea. Something to do with arginine. 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 Good. Interesting. I'm not sure why that was circled. Maybe there was... Oh, I see, because there's a difference. Gotcha. Yeah. All right. Uh-oh. All right. I think you're seeing what you're supposed to see. Can you see the boxes that I highlighted? I think you highlighted the same boxes.
You have to zoom in.
I'm hoping you're noticing that in the case of this particular amino acid, the amino acid arginine, there's a difference in the amount of arginine found in Beau's hair as a puppy compared to his hair as an older dog. But that those differences also appeared in mommy and daddy dog, right? That in dad, roughly the same percentage of arginine was found in dad, but in mom, there was more of this particular amino acid found than in dad or as Beau or in Beau as an older dog. Okay. All right.
What about
So,
tryptophan?
what about tryptophan?
Yeah. Isn't that the stuff in turkey?
No, that's a myth.
Well, I know it is a myth, but isn't that it just doesn't exist in turkey?
No, it's present in all animal protein, but it doesn't make you sleepy. But yeah, that it's a myth because it's found in everything, all proteins, then that means that all foods that have protein would make us sleepy. What makes us sleepy when we eat turkey is the fact that we're celebrating a holiday with family and friends and that we've probably been up really early and it's stressful and it's exhausting. So by the middle of the day, we're just done and we need a nap. All right, so there's some differences here. So to explore the effect of those differences, these hair samples were prepared for microscopic analysis. So now you took a piece of hair, really the whole hair follicles. Now we have to go back to the poor dog and pull out a piece of hair with the root at its base. And then we cut it, sliced it really thin. So somebody in the last class, imagine a tube of Pringles, and they cut the tube so that you could look inside and see the Pringles. That's what you're looking at in this picture. Okay. But these pictures were taken under the microscope. So what are you noticing about the structure of the hair itself? Are there any differences here? What do you notice? I'm going slow on purpose. I'm not trying to patronize you.
I mean, the shape.
Yeah, the shapes are different. And this is important because if I superimpose the data on top of the images of the hair, where the percent arginine, the percent of this particular amino acid was high, right, in the keratin protein that's found in the hair, the resultant shape of the hair follicle itself was more circular. And that is entirely different in the samples where the percent of arginine is low. I guess the hair looks more ovular, right? Flatter, squatter. Okay. So the point of all of this is that, again, if the sequence of amino acids is important in determining the shape, the structure, the three-dimensional form of the protein, the composition of the amino acids in that particular polypeptide is just as important. Because clearly, if the amount of a particular amino acid changes, and doesn't even change by that much, the shape of the protein would be expected to change. We don't see that the protein specifically here. Okay. But as a result of those proteins changing shape, the hair that contains those protein molecules changes shape. Let's pause there. You're wondering about this example.
Is arginine, will arginine always cause curly hair?
Turns out that this trend is true for all mammals, not just dogs. So if you have curlier hair, you have hair with more positively charged amino acids, like arginine as part of its composition. And the opposite is true for straight hair. Everyone's looking at each other going, who has the highest amount of arginine? All right, good. Good. So take home message, what's super important about this is that both the sequence of amino acids and the composition of amino acids will determine the structure of the protein that folds from the polypeptide that contains that sequence and composition of amino acids. Okay. So in the second half of the video that you watched for today, I hope that you watched for today, you were introduced to the way that we actually describe the structure of the proteins, or of these proteins. And I'm not going to re-lecture on this, but I want to give you a chance before we sign off today to practice with some of the ideas that you were introduced to in that video. So I'm going to scroll through here and get to the questions. So let's start with this one. This is a multiple choice question from a released AP exam. Think back to what we saw in the video. Feel like you're getting stuck on the word tertiary. It's not tertiary. It's a soft T, tertiary. Remember from the video that tertiary is just the term that we use to describe the three-dimensional structure of a protein. If you've answered the question and you want to take your model apart, you're welcome to do so. All right, we can take a look at how you did. Kind of a mixed bag, but certainly a majority is emerging and growing. Yeah, so R groups, remember R groups is just another way of describing the side groups. Okay, so the side groups are the parts of the amino acid that are actually interacting with each other as that polypeptide folds. Again, I'm hoping you're beginning to understand that the sequence and the composition of those amino acids, if that changes, of course, the side group sequences and the side group compositions will change, and that's going to influence the way that the protein folds. Wonderful. All right, let's do one more before we sign off. This one's a little bit longer, but it's a good place to wrap up. Of course, the question or the answers are on the next slide. Are we ready to see the answers? Trying to be sensitive to time. All right. So which of these folded polypeptides, right, proteins, best answers the question? How would that change in the polypeptide lead to a change in the protein? And for the sake of time, we'll look at how people have responded. Looks like A is the overwhelming choice, becoming more overwhelming. It's got to be A, right? And again, hopefully this question connects all the different ideas together before you leave. We've got the hydrophilic non-polar amino acid on the inside, the hydrophilic polar amino acid on the outside, and there's a structural change, right? The mutated protein with fewer amino acids looks different. It has a slightly different three-dimensional shape than the original protein does. All right, well done. Well done. Folks at home, that's it. You're welcome to sign off. That's our last question. I'm going to go ahead and spray off the surfaces here for the folks in the classroom. Thanks for all of your work today, your attention today, and I will see you in a couple of days.
Send it to your work. That's what I'm gonna your house. Your house. Thank you.
There'd be 2 people in the hall and 4 people in the hall.
Liam Naylor to the counseling office. Liam Naylor to the counseling office.
It's wet. Good to go. Okay. Otherwise, it's dry. No, we got that.
Is Abby though? I don't know. Staying online? Can I run to the restroom?
me get everything reset for you folks. Give me a second here. There we go. There she is. Uh-oh. My speaker go off. All right. Well, hi, everybody. I feel like something shifted today. Am I misreading the room? Am I misreading all of these classes? Has something set in that we might not even be aware of, but is happening nonetheless?
The vibe does feel different. Maybe just midweek fatigue.
Okay. Well, I'm going to try to take it easy on everybody today and really just do some practice and confirmation of ideas that you were introduced to in the videos this week. No homework for Friday other than test corrections, which aren't even due till Monday. So maybe take some time for yourself over these next few days, at least in respect to this class. Take a break, take care of yourselves. Know there's no final exam to prepare for. I'm sorry, but I'm still struggling with this notion of giving final exams. I can't. I don't know. You struggling with that too? Am I just a crazy person? The utility, the usefulness, the purpose. I don't know. Okay, sorry. Sidetrack. Plan for the day. You might recall about 6 weeks ago, I asked you to fill out a really short survey. There's 5 questions on it, I believe. It's the Mindful Awareness Attitude Survey. Really, it's a way to check in. So it's another form of check-in, honestly, to see how your awareness of your thoughts and feelings are influencing what you do in the present moment and how you react in the present moment. So I've asked you to fill out the survey in the past. I was hoping you'd be willing to fill it out again as our check-in this afternoon. So hopefully you can click on that link directly. If not, just let me know. Sorry.
Could you put the code back up?
Yep.
Just like cover.
I know. I wish I could get it to move around. So if you are willing to fill out this brief survey, just a reminder, it's 100% anonymous. I am not collecting your email address in the background, so please be honest. Just trying to monitor submissions, giving people a chance to respond if they'd like. Thank you for doing that. If you're curious, I'm really trying to find out if these check-ins that we do are having an effect, are having an impact. And of course, I'll never be able to argue like for this causatively, that this led to that. Right? This is still a correlation I'm trying to establish, but maybe once everyone has submitted their responses, I can show you just kind of globally how you folks are responding. There are some patterns that are emerging for some of the questions already, but I just wanted you to know that we're doing these check-ins for a reason. We want to know if they're having an effect, and I really appreciate you taking some time today to provide your feedback. Okay, on to the biology for the day. So you've watched a couple of videos now on protein structure and function. So today is going to really be about protein structure and function. But I thought before we go and practice with that information, we might go back a bit and see how what we're doing now connects to what we have done in the past. So here's a picture of a really simple polypeptide. What's being shown in the image? Which answer choice makes the most sense to you? AP multiple choice question.
Can you hear me if I talk? Hey.
All right, look at that. This is actually all 3 classes today already. So very well done. Of course, this is a hydrolysis reaction. We're taking that polypeptide apart. We're adding water and breaking, in this case, we can even highlight it or circle it. We are breaking this peptide bond, that covalent bond, and by adding water across that bond, regenerating the 2 functional groups. So we've got our hydroxyl that's being regenerated as part of a larger carboxyl group, and then this hydrogen is being regenerated as part of the larger amino group. Beautiful. Very nice. You are so smart. I agree. All right. So on to the new-ish, but really we're going to practice with what you already have seen. So today's all about proteins. So a reminder that if we're building a protein, we're building a protein from the monomer, the amino acid. So as we think back what we've done over these last few class periods, if we're building a fat, a lipid, we're building the lipid from fatty acids and glycerol. If we're building a carbohydrate, we're building it from monosaccharides, probably glucose is one of those monosaccharides. But if we're building a protein like we are today, we're going to be building those molecules from amino acids. And really, there are 20 different amino acids. You need to memorize the names and the structures, the 3-letter abbreviations and the 1-letter abbreviations for the next exam, which will be now. So go. He says sarcastically, and yet. No, these amino acids all pretty much are the same when it comes right down to their structure. What makes them different from each other are their side groups. I think we did a little bit of this the other day with a practice question. And yes, there are 20 of them, and no, you don't have to memorize their structures, nor do you need to memorize their properties. Either a chart like this would be provided to you, or enough information would be provided to you in the question to help you figure out the property of the amino acids that you're working with. So what we want to do today is put together a model, a really simple model of a polypeptide. So unlike in the practice question that we started class with a moment ago, we're not going to take apart a polypeptide, we're going to put it together from a set of amino acids. But remember, these are dehydration reactions, right? Water is going to be a product, and we're going to generate a new bond between those amino acids. That bond has a name if you are interested, because of course we need to memorize the name of that bond, the peptide bond. Don't you wish if a teacher ever told you to memorize something, you could freely and openly ask why? Do you know that you can? Seriously? You know that no is a complete sentence? No, I'm not going to memorize that. I have better uses of my brain power. Just saying, something to think about. I wonder what would happen if we used that complete sentence more often. So, short story long, you don't need to memorize the name of this bond. It does have a name, but it's really just a covalent bond, right? That's really what's most important if you're going to remember anything. These aren't ionic bonds, they're not hydrogen bonds, they're not London dispersion forces, they are covalent bonds. Okay, so here's how we are going to model this. And folks at home, I have a virtual equivalent of this for you, but bear with me while I get people here in the classroom set up. So what we're going to do is we're going to build a really simple model, not of a protein actually, but of a polypeptide, of a linear chain of amino acids. Because in fact, a protein is a folded 3-dimensional polypeptide. So we've got to build the polypeptide first before we can fold it into the 3-dimensional protein, okay? So what I am going to do for the people here in the classroom is give you a cup of push pins. Hope your hands are clean. Although, I think it's really just hygiene here at this point. Our masks are far more important than our hands. Unless you're like sticking your fingers in other people's mouths when I'm not looking. Is that what's happening? I smell a TikTok trend. Mr. Grisher said that for homework, I would get an A+ if I stick my dirty fingers in other people's mouths. By the way, I'm recording all of this for YouTube posterity.
When you go TikTok viral.
Maybe I will. Maybe I should start it. I have 0 followers.
Do you have a TikTok?
No. I like books. All right. So everybody has a little cup of push pins and this piece of foam covered wire. So what I'm going to ask people here in the classroom to do, and folks at home, you can just kind of pause for a moment while they do this, is insert your push pins into that foam covered wire in a way that it is evenly spaced. It's not going to be perfect. Don't worry about that. And of course, this really is an exercise in natural selection. So if you find yourself poking yourself again and again and again and again and again, well, nature's doing its thing. Thank you. Thank you so much. Nature is telling you something. All right. I think I'm losing it. In fact, while I'm doing this, folks at home, what are you seeing on your end? Are you seeing the foam covered wire? Or are you back to the agenda slide? Could you let me know real quick?
The foam wire.
Excellent.
Good.
Excellent. Good. So this is not going to be perfect, and for the sake of time, try to be as efficient as you possibly can. You don't want to make Abby angry. She's waiting so patiently. With Joshua and Joseph too. If you find yourself with some leftover space, big deal. Don't take everything out and then put it all back in again. There's not one correct answer to what we are doing right now. You cannot do it wrong.
What is this supposed to represent?
This is the polypeptide. So if I were to go back just a little bit, this is that linear chain of amino acids. Kind of over here. Yes, I'm going to show you what that is in just 1 second. Yes. Sorry, I thought you meant the overall.
No, we're just making this with no explanation. Just for
Hey, you never know. If I've truly lost it, maybe I'm hoping that you're all going to bleed slowly as you impale yourself on tiny pins. All right, things are looking good here in the classroom. Folks at home, thanks for pausing there for a moment, because yes, there is a method here that we're trying to utilize. So the idea behind this is that each of those push pins represents a different type of amino acid side chain. And this table that I'm hoping you can see on the screen now is what I'm hoping everybody can see. So the blue push pins represent positively charged side chains. These are basic side chains that picked up a proton from solution. Red push pins are acidic. They've lost a proton to solution, so they have a negative charge. Yellow are the hydrophobic nonpolar side chains, kind of like the methyl group that we saw the other day. And then white are polar or hydrophilic side chains, and the cysteine is green. Okay. So because the rest of the amino acid is the same, no matter what type you're looking at, the push pin is just the side chain of that particular amino acid. Okay. So what you have now is a polypeptide that consists of 15 amino acids all linked together. Their side chains are the parts that you can see via the push pins. So what we're going to do is take that polypeptide and fold it into a three-dimensional protein, but we're not going to do it randomly or stochastically. We're going to need to fold that polypeptide according to some simple rules of chemistry. Okay? The first assumption that we're going to make globally is that as our polypeptide folds, it's folding in water. which shouldn't surprise you too much because everything in biology happens in water. Okay. So what we need to have happen is those non-polar amino acids, the yellow ones, need to be buried on the inside of the protein because they want nothing to do with water. They don't interact with water at all. But the polar amino acids, right, the white ones, they can be on the surface of your three-dimensional molecule. The red and the blue, the positive and negative side chains, well, we want to get them into a position where they are bonding, where they're interacting with one another. And then finally, we want the green push pins, a special amino acid called cysteine, to be able to be close to one another because they actually can form a covalent bond with one another. So go ahead and see if you can fold your polypeptide into a three-dimensional structure following these rules. Folks at home, I have done this work for you, and I have some questions for you to consider on the next few slides. So I hope you can see this folded polypeptide on your end. If you can't, please let me know. But the only rule, if you're doing this in person, is you can't pull the push pins out and move them around. You have to fold the polypeptide according to the rules. One falls out, won't you? So if this were to be happening inside of a cell, it would be happening on the order of microseconds. So as an intelligent, sentient being, you should be able to do it on the order of nanoseconds. He says sarcastic. It's tough, right?
It's a lot harder than I thought it would be.
Yeah. But just as, if you take a moment to just kind of hold up your model and compare it to the models around you, what are you noticing? Some are more structurally sound than others. It's 10 points off page.
I'll raise my grade by doing the open autopsy.
Well, or you could bring in tissues for extra credit.
Do they have to be clean or dirty?
Preferably dirty. You know what? Give me a mix. But don't tell me which ones are which. Would it still be Russian roulette if it's tissue roulette?
I mean,
It's just the flu, folks. Don't worry about it. It's not real. COVID's not real. It's just the flu. People actually believe that?
yeah, probably. Until you get it.
Even then, apparently there are people on their deathbeds in hospitals that are on camera now saying, "It's just the flu." Uh-huh. Sorry, I heard that on the radio yesterday. And I thought,
So sad.
I heard someone had like, I don't have the vibrational frequency to get COVID or
Ooh.
something.
Oh, I saw that.
I like that. Is that trending on TikTok too? Along with dirty tissues?
It was on TikTok, actually.
Yeah, I don't know if it's trending, but it's
Oh.
definitely something that came out like
Definitely
pretty
trending.
I taught her that. I love that there are like millions of TikToks, yet somehow we managed to watch the same ones, right? I don't know how it happened, but it does. Frequently.
Well, everyone's like, did you see that TikTok? Yeah, we all saw it. The same one.
Yeah.
It's crazy is no matter what you're watching or how often you watch it or how you watch it, like as a science teacher, I just feel like I've failed society miserably. Like your resident energy frequency is such where you can't contract a virus. Like where did that come from? Or like 5G technology is going to cause like disruptions to your. Shut up! It's a radio frequency. What are you talking about? So one of the. Go ahead.
I went to like this holistic medicine person, and they tried to like. I like was not buying into it. My mom was like, "Do it. It's like just try it. Maybe it'll help or whatever." And the woman like did these like weird things to test what like I was like having aversion to, and then she took this little plate and this water, and she put the water on one thing and pressed a button that said copy, and then moved to the other side of the plate and said paste, and it was supposed to like reverse the frequencies of the atoms in the water, and I was like, that's just not possible. Like, I don't know what you're trying to tell me. Like, that's just not possible. And like, people like pay good money to go see this person, and so like, I just don't understand. Like, how do you believe?
Wait,
that?
why did you go there?
Because I was having stomach problems, and my mom was like, "We're just gonna try everything."
Reverse the atoms in your stomach.
She like literally and was like, "You need to do these droplets of water that have the inverse frequencies every 3 hours, and you'll be cured." And I was like,
Do you still have stomach problems?
"Yeah, oh."
Had you said no, we were like, really? Sign me up.
Stomach problems.
You should go there if you get from.
Yeah.
All right. So, folks at home, what everyone here in the classroom has built is a folded protein. But before we got a little sidetracked there, but understandably so, I think I led the sidetrack train. Everybody's three-dimensional structure is different. Okay. So, what made those structures different?
The locations of where we put our thumbtacks or like the amino acids were.
Exactly. So everybody started with the same length of tomato wire, and everybody actually started with the same push pins. But the order, the sequence in which you inserted those push pins into that foam-covered wire ultimately determined how that polypeptide would fold into the protein. And that's really the first takeaway here for today, that the sequence of amino acids in a polypeptide determines the structure of the folded protein. Okay. So before we go on to the next variable that influences protein shape, is this bringing up any questions for you? Folks at home, I hope you had a chance to play around a bit with these drag questions. All right, so before we go on to the next example, let's just reflect and confirm our understanding. Take a moment, read this AP free response question, and take a couple of minutes to respond with your best answer. Anyways, you've already modeled the prediction. You don't necessarily respond to that part of the question. So what would happen to your protein structure if you were to put the polypeptide into a non-polar environment? Think oil, think fat. Unsaturated or saturated, it doesn't matter. Those are non-polar molecules. So when you're ready, we're going to try this. I hope that it works. Folks here in the classroom, you can just turn to somebody close to you and share your answer with one another. Folks at home, I'm going to put you on mute so nobody will be able to hear you here in the classroom. But hopefully you'll take yourself off mute just for a moment and share your answers with one another. There's only 3 of you, so hopefully you're willing to do that.
Hey.
Hello. So I said the protein would likely fold upon itself and take up less volume if it were to be put in water.
I just said that they would fold up. Yep. Okay.
Same. And then for the nonpolar environment section, I said it would probably do the opposite and maybe unfold rather than scrunch up.
Mm-hmm.
We got this
I
version.
said it.
Hmm?
It sounds right enough.
All right. So I think the conversations have come to a close here in the classroom. Let's take a look at your responses and see what you've come up with. Yours are going to be towards the bottom here. They would fold in a nonpolar environment. That's true. Folded outward. Now we're getting someplace. Well, if the polypeptide was in a nonpolar environment, we expect the nonpolar side chains to be on the outside of the polypeptide chain of the protein, and the polar to be on the inside. So it's the opposite of this answer here. Fold inwards, fold outward, fold outward. Yeah, there we go. Now we're getting there. So the idea here is that we started, right, with the assumption that our polypeptide was folding in water. Okay, and we're coming back to some chemistry background knowledge here.
Is water polar or not?
Water is definitely polar. Yes. Yes. So water as a polar substance does not interact with non-polar substances. If you need to remember that, an easy way to remember is like salad dressing, vinaigrette salad dressing. Something we probably have been exposed to at some point in our lives. That the oil in salad dressing is non-polar, it's hydrophobic. The vinegar, it's 95% water, 5% vinegar, is polar, it's hydrophilic. So if you let salad dressing sit out, it's going to separate into 2 distinct layers, right? A polar and a non-polar layer, a watery layer and a fatty layer. Those 2 substances do not mix, and if you're going to go down the rabbit hole of, yes they do, I just shake the salad dressing. Okay, put the bottle down and let it settle and watch the 2 layers separate again. Okay. What if I add an egg? Yes, those are emulsifiers. You can get polar and non-polar substances to mix with each other if you add something that allows them to interact. That's not what we're doing here, right? We're starting with a polypeptide. We're letting it fold in water. The polar side chain should face out. The non-polar side chain should face in because the non-polar side chains cannot interact with water. If instead we were to allow that polypeptide to fold in a non-polar environment, everything gets flipped, right? Now those non-polar side chains are going to face outward, and the polar side chains are going to try to get as far away from the non-polar environment as they can. They're going to be found on the inside of the protein. Okay. So, does that help? Well, let's just look at one other factor that we know influences the shape, the structure of proteins. And I believe you were introduced in the second video to Bo the dog, but you were told you're going to learn more about Bo in the classroom. All right, so here's Bo's family tree. Mommy and daddy dog up top, Bo and his siblings down below. What do you notice about Bo's fur? It's the cutest, a subjective determination, but fair.
It's like thicker than that of his parents, and then it's straighter and then fluffier because just now.
Yeah, it's straighter, it's fluffier, it's thicker, right? If you look at daddy dog, that old mutt, he tends to have curlier hair. But what's interesting is if you look at Bo not as a puppy, but as a slightly older dog, who does Bo more closely resemble? Dad. And his siblings? Look like mom. So when scientists observe this, and this is years ago now, but it still holds true, and it holds true for other mammals as well, mammals including humans. They wondered, okay, well, wait a minute, what makes hair in dogs, in this example, different from one another? So they isolated that hair from the dogs, and hair is mostly protein. It's actually dead as well. If you're investing money on vitamin-enriched shampoo for healthy hair, I don't know how you revive something that's dead, but maybe you believe in zombies, so that's fair. So, add those vitamins to your dead hair with the hopes of making it healthier. And while you're at it, when you have dinner tonight, rub your dinner on your scalp as well with the hopes that you will provide it with some nutrients to grow thicker and more luxurious tomorrow morning.
Wait, so for vitamin shampoo, it actually does have vitamins in it, but they just don't do anything? They kind of just wash off?
Nothing. They go down the drain. Yep. If that were true, we would rub all sorts of things onto our body in an attempt to gain nutritive sustenance.
Instead of like vitamin-rich like hair products, if you like, I know there's so many like marketing things for like hair vitamins. Take this and get long, luscious hair. Like, do those have like? Does that do anything?
It gets us quickly down into the weeds of biology, but I would much rather you eat food that contains those nutrients rather than take a supplement that contains those nutrients. And the short answer to that conundrum is that lots of nutrients are absorbed better with food because they come with other molecules that make the absorptive process more efficient. Simple as that. If you ever wondered why, for example, milk is fortified with vitamins A and D, it's because vitamins A and D are fat-soluble vitamins, and milk, even skim milk, has a little bit of fat in it, which makes it more likely for those molecules to dissolve, and as a consequence to be absorbed by our body once they're ingested. All right, but quickly here, back to Bo and his family. The hair samples from all of the dogs, Mommy, Daddy Bo, and his brothers and sisters, I don't know their sex, I apologize, were collected. And since hair is mostly protein, one type of protein called keratin, in fact, the scientists decided to figure out what the amino acid composition of the protein in that hair happened to be. And these are the results. So quickly, can you highlight any differences that you see between either Bo as a puppy and Bo as an older dog, or Bo and his mom, or Bo and his dad, or Bo and his siblings? You could circle the differences, you could highlight the differences. Just quickly scanning the data and highlighting any differences that you notice. If you're wondering, ND is short for not determined, which really in this case just means that there wasn't any of that particular amino acid in the protein, in the keratin protein that makes up hair. right? So my hope is that you noticed these differences here. I'll draw on the slide so that you can see them. There's a large difference between the amount of this particular amino acid, arginine. Oh, and by the way, make sure that you can spell that correctly. Spelling counts, right? There's a pretty large difference between the percent of this particular amino acid in the protein within Bo's hair compared to when he's a puppy and an older dog. And that these percentages roughly match up to what you see in Bo's parents. So daddy dog has a lower percentage of this particular amino acid, right? Mommy has a slightly higher percent of this amino acid. So with that in mind, scientists wondered, okay, well, what might that difference be responsible for? And that's where I think things get really interesting, because they decided to take these hair samples from all of the dogs and prepare them for microscopic analysis. So somebody in an earlier class had a really good analogy for this. They imagined a Pringles can, a cylindrical can, that's your hair. And they cut the cylindrical Pringles can in half, and they looked inside of the Pringles. That's what you're looking at in this image, the inside of a hair. Okay. What are you noticing?
The curlier hair is more like disc shaped.
The curlier hair, so the hair found in older Bo and daddy, is more disc shaped. Somebody said ovular or kind of squat. I like that word squat too. Compared to these more cylindrical, circular forms. And it turns out that if you have a higher percentage of arginine as part of the keratin protein that makes up your hair, you tend to have straighter, more cylindrical hair fibers. But in contrast, if you have a lower concentration of this one particular amino acid, the composition of that amino acid in the keratin protein is different, and the hair looks different under the microscope. And looks different to the naked eye. Higher arginine, curlier hair. Less arginine, straighter hair.
So is there a way to like put that into a supplement to
No, because really what this comes down to, and maybe you learned a little bit about this in middle school, right? The proteins that we're talking about here, keratin, this example, it's made according to a set of instructions that resides in your DNA, in your cells. So those proteins are always going to be made in the same way, either lots of arginine or a little bit of arginine. And this actually, I made this slide. I don't know that we have time to delve into it, but it's determined on the based on the genetic background of your parents and what you inherit from your parents. So Bo's instructions for making this protein are such that he makes less, or he makes keratin protein with less arginine compared to his siblings or his mom that makes the keratin protein with more of this arginine. Okay. Sorry, do I have that flipped? I have that flipped. Straighter hair.
straighter hair is more.
Like I said, my brain is going crazy here. Straighter hair has more arginine. Curlier hair has less. Okay. So take-home message from all of this is that not only does the sequence of amino acids matter, but the composition of the amino acids within that polypeptide matters in terms of how that protein folds, the shape that it will possess, and how it functions. In this case, the function isn't really anything more than the shape that the hair takes on the outside of the organism. Okay. So my hope is we can do one more question before everybody signs off or takes off. And if I can get to it quickly enough, it connects some of the different ideas that we've tackled today. So in these last couple minutes, this is our exit flip for the day. Just give yourself a moment to read this question. If you want to take your model apart while you're doing it, feel free. Okay for the answer choices? They're on the next slide. If I change the composition of the amino acids, I remove some of the hydrophobic ones, how's the protein going to change? All right, for the sake of time, I hope you have your answer in there. It's A, right? The hydrophobic, the nonpolar side chains are still on the inside. The hydrophilic are still on the outside. The polar are still on the outside. But the shape is different. Subtle, but different. All right. Thank you, everybody.
Bye-bye.
Bye-bye.
Abby, bye.
Bye-bye.
See you later.
Yeah.
Liam Naylor to the counseling office. Liam Naylor to the counseling office.